Hi, today I want to talk about deep generative models used in protein engineering. The goal of using these models is to learn the underlying data distribution for tasks like learning meaningful representation of sequences, sample sequences that haven't seen before, and assign high probability to desired sequences. Within these four boxes are the applications of generative models. The first is initial training. The blue encoder will encode sequences into their low dimensional vectors in embedding, and the generative decoder will reconstruct the sequences by using those vectors in embedding. The training of this model is trying to make sure the embedding holds the valuable information about the sequences and exclude the noises. Often, the generative decoder can be replaced with a predictor for tasks like classification or prediction on protein activity properties. And the loss can be calculated and it can backpropagate to parameters in predictor and encoder in order to maximize the model performance. And this is also called fine-tuning on downstream task. And C is just taking the training embedding applied with a generative decoder to generate new sequences. And the last one, optimization, is using the decoder to generate optimized sequences by applying reinforcement learning. So by assigning weights to training sequences, the model will reward the neurons that make right decision by increasing the weights and weight bias within that neuron. And this paper also talks about the three very popular generative models right now. The A is variational autoencoder. The encoder will learn the probabilistic distribution of data and store those distribution as mean and variance in the latent space Z. And the decoder will generate outputs by sampling the distribution in the latent space Z. So the objective of this model is to maximize its P of X, and the direct integral of probability density function is intractable, so that this model actually only maximizes the lower bounds of log P of X to get the maximized P of X. And the B is generative adversarial networks. The generative part comes from the generator, which generates fake sequences, and it has a second network called discriminator. Its function is to differentiate between the fake sequences and the real sequences. So these two networks kind of like compete each other and help each other to do the task better. This model has a minimax objective function. The discriminator has a maximize objective, so that d of x is close to 1 and d of g of z is close to 0. And for generator, it has a minimize objective, so that the d of g of z is close to 0, because the fake data z wants to fool the discriminator by thinking the fake data is real. And the last one is autoregressive model. The generation is one by one, each token, and it's also conditioned on all previous tokens, like the here shows. So the objective of this model is trying to maximize P of X, and P of X is equal to the product of all the probability of X at each position, the amino acid at each position conditioned on the previous amino acids. I highly suggest to read this paper because they have a lot of actual examples by implementing these models.